हेलो बच्चों, वेलकम टू SPSVERSITY. In the previous session, we discussed about micromolecules and macromolecules. In that we discussed about in micromolecules the sugars, amino acids, the nucleotides. Whereas in macromolecules we discussed about polysaccharides, proteins, nucleic acids. Okay. In this session we are going to discuss about the nature of bonds in polymers. So polymers means specifically macromolecules. So macromolecules are nothing but the combination of micromolecules. So these micromolecules are linked by bonds. माइक्रोमोलिक्यूल्स के बीच में क्या रहता है कि बॉन्ड्स. ये बॉन्ड स्पेसिफिक रहता. फॉर एग्जांपल इफ यू टेक प्रोटीन्स, so in proteins there are amino acids. The bond present between the amino acids is called peptide bond. That is, during protein synthesis, peptide bond is formed between amino acids. In the same way, if we take polysaccharides, polysaccharides are made up of monosaccharides. Polysaccharides are complex sugars which are made up of simple sugars. They are monosaccharides. ये मोनोसैकेराइड्स के बीच में ग्लाइकोसिडिक बॉन्ड रहता. Okay. And when you come to nucleotides, when you come to nucleotides, there are again three components. That is, in nucleotides, there is a nitrogen base, a pentose sugar, and a phosphate molecule. So it is a combination of three components: nitrogen, sugar, and phosphate. नाइट्रोजन और शुगर की फॉस्फेट के बीच में ग्लाइकोसिडिक बॉन्ड रहता. दे आर लिंक्ड बाय ग्लाइकोसिडिक बॉन्ड. और शुगर और फॉस्फेट के बीच में क्या रहता है कि फॉस्फोडाइएस्टर बॉन्ड. So two nucleotides are linked by phosphodiester bonds. Nucleotides are linked by phosphodiester bonds. So here you can look into this. So this is, you can see a glycosidic bond. Okay. So here these are amino acids. These are between sugars. Okay. Between sugars there is glycosidic bond, that is in polysaccharides. And when you come to proteins, between the proteins there is peptide bond. That is, in between the amino acids there is a peptide bond. And when you come to nucleic acids, in nucleic acids में the basic blocks nucleotides रहता. A nucleotide और nucleotide के बीच में phosphodiester bond रहता. So this is one nucleotide. This is one nucleotide. So from phosphate, the sugar, to nitrogen base, this is one nucleotide. Between the nucleotides there is a bond, that is phosphodiester bond. So ऐसा polysaccharides में glycosidic bond रहता. प्रोटीन्स में पेप्टाइड बॉन्ड रहता. और न्यूक्लिक एसिड्स में न्यूक्लियोटाइड्स के बीच में फॉस्फोडाइस्टर बॉन्ड रहता. After studying the types of bonds, so next we are going to study about the concept of metabolism. Metabolism बोले तो क्या है? So all the chemical reactions that occur in a living organism, means that occur in a cell, which constitute metabolism. मेटाबॉलिज्म में दो टाइप्स हैं, एनाबॉलिज्म और कैटाबॉलिज्म. एनाबॉलिज्म इज ए कंस्ट्रक्टिव प्रोसेस. So constructive means from simple substances, complex substances are formed. Like for example, if you take protein synthesis. Protein is a complex macromolecule which is formed by combination of amino acids. इसमें बहुत सारे amino acids रहता. ये amino acids के बीच में peptide bond रहता और peptide bond और amino acids combinedly it is called protein. Amino acids are simple one, protein is a complex one. Catabolism, it is quite opposite to anabolism, where complex substances are broken into smaller substances. Like, for example, if we take digestion of protein. So protein digests होने के लिए, when protein is digested with the help of proteases, the enzymes which will digest proteins, they are called proteases. A proteases finally after digestion of proteins will get at the end amino acids. That is from complex substances, that is from protein amino acids. The same like if you take polysaccharides. On digestion of polysaccharides you will get monosaccharides. So this is called catabolism. So metabolism में दो types रहता. One is anabolism, another one is catabolism. Anabolism is formation of complex substances from simple one. Catabolism means formation of simpler substances by breaking down of complex substances. Okay. ऐसे बहुत सारे रिएक्शंस इन ए सेल होता है. ये पूरा रिएक्शंस को क्या बोलते हैं कि मेटाबॉलिज्म. So what is this living state? Okay. A living state में वर्क परफॉर्म होता है. So it is a steady state and a non-equilibrium steady state. Our body will be trying, living organism will always try to reach to equilibrium, but it never reaches to equilibrium. So this work is called living state. So this work is performed continuously. It's not it's a continuous process. All living organisms will do this work continuously but cannot reach to equilibrium. Always closer to equilibrium but they cannot reach to equilibrium. It's a constant effort to reach to equilibrium. Okay. To reach to equilibrium, this constant effort requires energy. Okay. This constant effort requires energy. Okay. In this living state, to reach to equilibrium, energy is required. Okay. As energy is required, so this energy comes from respiration. So respiration is one metabolism from where energy is produced. Okay. So here where we can come to this living state and metabolism, both look synonymous. Okay. So without metabolism, there is no living state. Okay. Both look like synonymous, but they are not synonyms. So living state is a condition where they perform a continuous work to reach to equilibrium. But metabolism is the type of the work, whether it is anabolic or catabolic. So without metabolism, there is no living state. So for achieving living state, metabolism is required. Okay. So whatever the metabolic reactions are occurring, so these all metabolic reactions are catalyzed by enzymes. What are these enzymes? So enzymes are made up of proteins which will catalyze the chemical reactions. Catalyze means enzymes are nothing but biocatalysts. So they are commonly called biocatalysts. Okay. Whatever the reaction takes place, whether it is anabolic or catabolic process, enzymes are required. Taking from digestion to respiration, whatever it is, all the process which takes place inside the cell, all are catalyzed by enzymes. Most of the enzyme that is are proteinaceous, they are made up of proteins. But even in some cases like nucleic acids also act like enzymes. That is during protein synthesis. They are called ribozymes. So ribozymes means where nucleic acids will act like enzymes and perform the process. So इसको क्या बोलते हैं कि ribozymes. Okay. अब मैं देखेंगे केमिकल रिएक्शंस में क्या चेंजेस होता है. केमिकल रिएक्शंस बोले तो फिजिकल चेंजेस रहता, केमिकल चेंजेस रहता. Whatever it is, whether it is a physical change or chemical change, there will be a rate of reaction. Rate of reaction means the amount of product formed per unit time. Any reaction by the end of a particular time, the amount of product formed is called rate of reaction. Okay. So this rate of reaction is increased by enzymes. That is where enzymes are involved. इसको क्या बोलते कि catalyzed chemical reactions. Here the question is, if enzymes are not involved, the reaction will take place or not. The reaction will take place uncatalyzed. Whether the reaction is catalyzed or uncatalyzed, both are the reactions will take place. But what is the difference? Catalyzed reactions are faster than uncatalyzed one. That means the reaction can be faster, like 10 million times, accelerated by 10 million times. So enzymes will increase the rate of reaction. So enzymes will increase the rate of reaction. So they are called catalyzed or uncatalyzed. Okay. This is the difference between catalyzed and uncatalyzed reactions. So here you can see the formation of carbonic acid in the process of carrying carbon dioxide in the blood. During this process, carbon dioxide, 70% of the carbon dioxide is carried out in the form of carbonic acid. That is, carbon dioxide will combine with the water present in the blood, which will form a carbonic acid. So, which is catalyzed by an enzyme, carbonic anhydrase. So, which is catalyzed by the enzyme carbonic anhydrase. So, this enzyme will play an important role in the formation of carbonic acid. So, in the presence of enzyme, the reaction takes place, that is 10 million times faster than without the enzyme. If there is no enzyme also, carbonic acid is formed, but it takes very slowly. Uncatalyzed reactions are very slow. Catalyzed reactions are very faster. Okay. So this is the role of enzymes. So how do enzymes function? Okay. Enzymes are made up of proteins. Okay. Enzymes make careta. Enzymes have pocket-like structures or crevices. A crevices means substrate fit होता, substrate बोले तो क्या है? Substrate means the reactant. In chemistry, we call it as a reactant. That is, to proceed the reaction, enzyme has to combine with the substrate. So substrate has to fit into pocket of enzyme to form enzyme substrate complex. So पहले क्या form होता है कि enzyme substrate complex. ये enzyme substrate complex it is formed by combination of enzyme plus substrate. After this, substrate is converted into product. P means product. It is transformed into product through a transient state. After formation of product, enzyme and product are separated. So enzyme and product are separated. So पहले enzyme plus substrate forms enzyme substrate complex. Enzyme substrate complex, after transformation of substrate into product, again enzyme and product are separated. Here there is a change of substrate to product, but enzyme remains constant. The structure of enzyme, or chemically the enzyme remains constant. It doesn't undergo any changes except the substrate to product. Okay. So during a catalyzed reaction, so there is no change in the structure or chemically enzyme. Only substrate is transformed into product. How these enzymes will transform the substrate into product? What is this? What will happen? What enzymes will actually do? To understand this, we need to discuss about concept of activation energy. What is this activation energy? This is an external energy to start a chemical reaction. So, activation energy is an external energy required to start a chemical reaction. Okay. So, this activation energy, which is required to start a chemical reaction. So, if you look into the graph, so this is energy. This is reaction coordinate. Okay. Here, if you look into this picture, into the graph of this, so this is activation energy. When there is no enzyme, it is very high, it is in peak. Okay. In the presence of enzyme, the activation energy level is low. So this is activation energy with enzyme. This is the level of activation energy with enzyme. This is the level of activation energy without enzyme. So here, what enzymes are doing here? So in the presence of enzyme, the activation energy is lowered by enzymes. It is lowered by enzymes. As the lower this activation energy, the reaction proceeds very faster. Okay. What these enzymes will do is they will decrease the activation energy levels. Due to this, the reaction will proceed faster. As high level of activation energy will slow down the reaction. It takes very, very slowly. So enzymes, what they will do is they will decrease the level of activation energy. So this is what enzymes will do in a catalyzed reaction. They will decrease the activation energy. So here enzymes functioning, how they function. Okay, we studied that enzymes will decrease activation energy. Okay. Based on this enzyme functioning, there are two hypotheses. One is lock and key hypothesis, another one is induced fitment. What is this lock and key says? Lock and key means just like a lock. For every lock, there will be a specific key. When the specific key is introduced to the lock, it will open. So here if you take this, for every enzyme there will be a specific substrate. So here seeing pocket-like structures, इसको क्या बोलते हैं कि active sites. Okay. So the shape of active sites is similar to the shape of substrates, which will fit into them. Other substrates which doesn't fit into this means they don't have the similar shape. That means, for example, if you take substrate is like a key. Enzyme is like a lock. So every lock will have a specific key. Without that key, the lock cannot be opened. So in the same way, the enzyme will have a specific shape of active site into which the substrate will attach. Okay. Where after attaching of substrate, the reaction will proceed where finally product is formed. Finally, the product is formed. So lock and key says the rigid shape of substrates and active sites. But what about induced fit model? What it says? Induced fit model means it says the enzyme active sites are not rigid, they are flexible. Okay. According to the shape of substrate, the shape of active site of enzyme will slightly change. Not too much broad, a slight changes will take place according to the shape of the substrate. So what induced fit model says is the active sites of the enzymes will slightly change according to the shape of substrate. So it is a rigid structure, whereas it is a rigid structure in lock and key, whereas in induced fit model it is a flexible one. Okay. So this is the hypothesis which says the functioning of the enzyme and substrate. Okay, we have studied that enzymes are made up of proteins. So what are the factors affecting enzyme activity? Enzyme activity is controlled by many factors like temperature, pH, concentration of the substrate, concentration of enzyme. These all will affect the functioning of the enzyme. Okay. As enzymes are made up of proteins, so proteins are affected by the temperature changes. Okay. So enzymes are active with narrow range of temperatures. Okay. The optimum temperature means where enzyme activity is at a peak, it is called optimum temperature. Okay. So here if you look into this, the enzyme functioning here, it is at 40 degrees, it is at peak, it is called optimum temperature. Below this 40, the enzyme activity is low. After 40, the enzyme activity has decreased. That means here if you look into this, the enzyme activity will decrease below this optimum temperature and above this optimum temperature. Okay. So in high temperatures, the proteins undergo denaturation. So their foldings will change at high temperature. Okay. That's the reason why at high temperatures the enzymes cannot function. At low temperatures the proteins become low, that is the proteins will become inactive. Okay. That's how the temperature will affect the enzyme activity. So what is the effect of pH? Every enzyme will have a optimum pH. It is not same for all. Optimum pH means at a specific pH its activity is very maximum activity. Okay. All enzymes will not have the same pH. So it is optimum pH where the enzyme activity is maximum. If you take intracellular enzymes, intracellular means सेल के अंदर रहने वाले एंजाइम्स को क्या बोलते हैं कि इंट्रासेल्युलर. Intracellular enzymes mostly they are at optimum pH is at neutral pH. Okay. For example, if you take salivary amylase, which is secreted into buccal cavity. So this salivary amylase, it is active at 6.7, nearly to the neutral. When you come to activity of enzymes in the stomach, like pepsin, renin, these all are. Their optimum pH value is 1.6. Okay. So the optimum pH of all the enzymes will not be same. It will be differing based on the activity. For example, if we take the enzymes in the intestine, they work at alkaline pH. If you look at the enzymes which function in stomach, their activity is maximum in acidic pH. So like this, pH will affect the functioning of the enzyme. So one we studied is temperature and another one is pH. Okay. And when it comes to effect of substrate concentration, as we go on increasing the substrate concentration, the rate of reaction also increases. Okay. This is up to only to some extent. So here if you look into the graph, so here you can see there is an increase up to this point. But later on it has come to a constant state. Okay. So where the substrate, even though if we increase the substrate concentration, the rate of reaction has come to constant. What is the reason for this? Why? Because all the active sites of the enzyme are occupied with substrates. So all the active sites of the enzymes are occupied with substrate. So iske vaste, the rate of reaction is constant even after increasing the substrate concentration. So we studied factors like temperature, pH, substrate concentration, which has effect on the enzyme activity. The next one is competitive inhibitors, which will inhibit the functioning of the enzyme, which will decrease the functioning of the enzyme. Like there are competitive inhibitors. What is this competitive? Competitive bolite. Inhibitors or substrates look same. Inhibitors or substrates look same. Okay. So these inhibitors will compete with the substrate. Okay. That is symbol is the same. So instead of substrate, the inhibitor will attach. Due to this, the enzyme activity will get decreased. It is called competitive inhibition. There is a competition between the inhibitor and substrate. At the same time, there are another inhibitors like non-competitive inhibitors. This non-competitive inhibitors, instead of attaching to active site, they will not attach to active site. They will attach to a site called allosteric site, that is other than active site. Due to this also the enzyme activity will decrease. It is called non-competitive inhibition. So inhibition में दो types रहता, competitive inhibition or non-competitive inhibition. Competitive means where there is a competition between substrate and inhibitor. Non-competitive means where the non-competitive inhibitor will attach to allosteric site. Competitive inhibitor will attach to active site. Due to this inhibitors, the enzyme activity will be decreased, where the reaction, the specific reaction cannot be performed. So we come into the functioning of enzyme, how enzymes will function. Factors affecting the functioning of enzymes. Now we'll study classification of enzymes. So this classification of enzymes is done by International Union of Biochemistry, IUB, which has classified enzymes into 6 classes. So enzymes may change types, like oxidoreductases, or even then they are called dehydrogenases. Other name is dehydrogenases. So what will happen here? The substrate is either reduced or oxidized. So here you can see S means substrate, which is reduced. Instead, after reaction it is oxidized. Oxidized substrate is after reaction it is reduced. Transferences. What will happen here? The specific group is transferred from one substrate to other substrate. So here you can see in this reaction, to a substrate a group is attached. So this group is transferred to this substrate by the end of this reaction. They are called transferences. Hydrolysis, which will function in the presence of water. They will perform hydrolysis. Hydro means water, lysis means splitting. Like if you take the proteases, which cause the breaking of peptide bond, hydrolysis of peptide bond. The same like amylases, which cause isolation, the hydrolysis of glycosidic bond, breaking of glycosidic bond. Lipases, ester bond, the breaking of ester bond in fats. So they will function in the presence of water, which causes hydrolysis of bonds, which will break the complex components into simple components. Ligases, which will remove the groups from the substrate. Okay. They'll remove. Here you can see to X and Y there is a groups are attached. They are removed ultimately. Isomerases, iso means same. So they will convert one form of isomer into another form of isomer just by rearrangement of atoms. So one isomer is converted to transformed into its nearest isomer. इसको क्या बोलते कि isomerases. And the last one, ligases. Ligases means which will link together two compounds. Catalyze the linking together of two compounds. It is called ligases. So enzymes may change types: dehydrogenases, transferases, hydrolyses, lysases, isomerases, ligases. So depending upon the functioning, there are 6 types of enzymes which is proposed by IUB, International Union of Biochemistry. Okay. Next, when you come to the components of enzyme, enzymes में क्या रहता? Okay, we studied the proteins. Along with protein part, there are other components also. इसको क्या बोलते कि cofactors. So these cofactors are non-protein constituents. They are non-protein constituents. So which make the enzyme more active. So cofactor is non-protein part and the protein part is called apoenzyme. So एंजाइम में प्रोटीन पार्ट को क्या बोलते हैं कि एपोएंजाइम और नॉन प्रोटीन पार्ट को क्या बोलते हैं कि कोफैक्टर. A cofactor में 3 types of cofactors. So prosthetic, the first one is prosthetic. And the next one is coenzyme. And the next one is metal ions. So 3 types of cofactors: prosthetic, coenzyme, metal ion. What is meant by prosthetic? So prosthetic is an organic compound, carbon-based one, which is tightly bound to protein part, coenzyme. Like the enzymes, if you take peroxidase and catalase, which will catalyze hydrogen peroxide. So here heme is the prosthetic. Coenzyme, these are mostly like vitamins. Okay. So these like NAD, nicotinamide adenine dinucleotide, nicotinamide adenine dinucleotide phosphate, which contain vitamin niacin. They contain vitamin niacin. Okay. Metal ions. Okay. So many enzymes require metal ions which are inorganic, which will form coordination bonds with the side chains of enzymes. Okay. Like for example, if we take carbonic anhydrase, which will form carbonic acid from carbon dioxide and water, and carboxypeptidases, in which the metal factor is zinc. So zinc is a cofactor of carbonic anhydrase and carboxypeptidases. So cofactors में prosthetic group, coenzyme, metal ion, 3 types of cofactors. So where the prosthetic group is tightly bound, which is organic. Coenzyme like vitamins. Metal ions like zinc, which is a cofactor of carbonic anhydrase. So these are the components, that is cofactor, different types of cofactors present in an enzyme. The non-protein part, the protein part is called apoenzyme. That's all for today's session. Follow us on SPSVERSITY. Thank you all. We'll meet in the next session.